Good morning, good afternoon, and good evening to all of you. This is Ginny LaBelle with Defined Health, and we welcome you to another edition of our Insight Series webinars. Today, Jeff Bockman, Vice President of Defined Health, will present From Pariah to Darling, Biopharma finally courts Immunotherapies. Defined Health is pleased to bring you these webinar presentations as a way of showcasing how we view and think looking forward about various critically relevant topics for the biopharmaceutical industry. Our viewpoint is the result of our long history of consulting with hundreds of pharma, specialty pharma, biotech, and investor clients. In these engagements, we bring our unique perspective, grown over 30 years of evaluating pipeline and inline opportunities by bridging our knowledge of drug development, business development, and commercial strategy across multiple therapeutic categories and technology platforms. I want to quickly call your attention to two of our in-person events happening in March of 2014. You can see the meetings up on the screen at the moment. Defined Health will present the 25th Annual Cancer Progress Conference in New York on March 4th and 5th. I encourage you to visit our website, cancerprogressbydh.com, to view the program, which includes world-class oncology luminaries from academia and industry, investors, and government. The agenda ranges from biomarkers, diagnostics, and big data to advances in treating brain and prostate cancer to antibody drug conjugates, gene and cell therapies, translational oncology, and pricing and reimbursement. Again, you can check the program out at cancerprogressbydh.com. Secondly, Therapeutic Insight by Defined Health will be held at EBD Group's BioEurope Spring Partnering Conference that will take place March 10th through 12th in Turin, Italy. Our 4 panels this year feature senior industry executives from pharma, biotech, and specialty pharma speaking about establishing proof of relevance in early-stage deal-making, cognition disorders across the CNS disease space, differentiating early in the autoimmune inflammatory disease space, and immuno-oncology. To learn more about Defined Health, our capabilities, and our upcoming annual Therapeutic Insight and Cancer Progress conferences, please visit our website at definedhealth.com or send us an email or give us a call. We'll be happy to tell you more. A couple of quick housekeeping notes. The slides will be up on our website shortly. We should have time for questions at the end of Jeff's presentation, but if we don't get to them, send them along anyway, and we'll follow up with you the next day or so. Thank you all, and enjoy the presentation. Jeff?
Great. Thank you. Thank you, Ginny. Thank you, everyone, for attending. So I think it was a little easier this time to come up with a title, given some of the changes that have happened over the past few years. And as we'll talk about, it's been an interesting sea change, clearly a very dramatic one at that. A few disclaimers. These are all based on our own thoughts and opinions and various sources of information that we have access to. My usual disclaimer about the various people that I am not, including Steven Spielberg. So there's me, and I've including now my colleague Mike Rice, who shares the oncology practice with me, who is not the captain from the movie Airplane. And of course, this idea of discriminating one person from another or self from non-self is inherent in our discussion of immuno-oncology. So I have the nice laureates here of Doherty and Zinkernagel staring at their reflection and seeing their MHC ribbons there. So I'm going to start with setting the context with the idea of kind of why is immunotherapy so appealing. I think there are a number of reasons that are scientific, clinical, but also commercial that are driving this. I'll move into then a little bit of some history here, and then take a look and dive into some of the more advanced programs that are currently going on, and then draw some broad observations about what's going on, at least on those advanced programs. So I think one of the things that is clearly important and on everyone's mind these days, although I think it does sometimes get overlooked, and it's something that cannot be overlooked in the increasingly cost-constrained, cost-conscious world that we are moving into, even here in the States, and that is that, by and large, we are not curing cancer. There are a lot of ways you can slice and dice the data, and there certainly are outliers in settings where patients are responding significantly and dramatically, and where they have had dramatic changes in kind of lifespan, whether we're talking about CML, which remains a poster child, or even where we're talking about cases like myeloma. But by and large, we are still stuck because of the fact that in most cases we're still diagnosing many of these cancers at the advanced stage, and therefore we're playing a game of catch-up and trying to outsmart a very wily competitor, being the tumor. So the incremental advances, while incremental can still be meaningful, particularly when one is gaining several months of life, that's clearly important at a personal level for the family and the individual with the cancer. Kind of in the broader perspective, and certainly in this, as I said, kind of increasingly cost-conscious perspective, this has tremendous implications in terms of how drugs will be approved, how they'll be used, and of course how they'll be priced, because it's not going to be a case where we can get approval and get wide usage with 2 weeks. I think that's obvious, but in many cases, where that break point will be remains to be determined. So one of these big unmet needs remains is how to significantly advance the outcomes, whether that's PFS or actually the golden rule of OS. So, part of this complexity here, whether it was in the days of purely of cytotoxics, whether it's the transitional days now of, in most cases, combinations of targeted agents, biologics and SMIs with cytotoxics in many cases, or even, alone or with each other, is we're still faced with a situation where the cancer inherently begins, or certainly at the time in the advanced cases that most patients are being reached at, with a tremendous heterogeneity and diversity, which just exemplified here from the NEJM article last spring in terms of just the intratumor heterogeneity, let alone any differences that may exist from one person to another person's tumor, and also not taking into account the heterogeneity of the context that that tumor is in, and how the microenvironment and the immune system, et cetera, vary from patient to patient, and the role that plays in terms of the original outgrowth of the cancer, as well as the ultimate response to certain agents, and ultimately the ability to control that cancer. And so this idea that no tumors are the same, that represents both opportunities and challenges. And one thing in a number of projects that we've been doing over the past year, where we've had a lot of conversations with luminaries, many leading KOLs, is we're hearing a lot of comments about the challenges of acting, the idea of moving from cytotoxics, which acted broadly, but clearly they had a lot of problems because of that, to the idea of focusing on very narrow subsets of patients and specific changes, whether those are driver mutations or not, and that this too narrow focus may at times be somewhat reductionist, but even if it's not, there are additional challenges that we face, because that cancer is not simply going to sit back and take our therapeutic interventions like someone with hyperlipidemia or hypertension, but it's going to fight back. And so the Darwinian evolution that you can see on the left, or the very specific examples in the schematic of some of the types of changes that undergo in terms of resistance to classic EGF inhibitors, et cetera, and the various mechanisms in terms of activation through MET and a variety of other things, highlight this issue of the plasticity and the acquired resistance. There was a huge session at AACR and at various other major meetings, because this is a major problem, because even with these very targeted agents, even with these ALK inhibitors or BRAF inhibitors, even when we're getting, durable responses there in small number of patients, but then ultimately, there is breakthrough and resistance. And so the idea is that, maybe immunotherapy, because of the nature of the immune system itself and its ability to deal with diversity and its own inherent diversity, that maybe if that immune system can be reawakened, maybe if that immune system can be harnessed, maybe if that immune system can be brought in exogenously or the brake taken off, that, perhaps this type of more conceptually broadly acting approach might be a great advantage in the face of such heterogeneity and plasticity and diversity and acquired resistance. Now, of course, one of the challenges there in thinking about how one does immunotherapy, and this applies and is inherently related to the types of immunotherapy approaches that one will deal with, is how do you do it? Do you think about historically, is there any reason to believe that it is better to deal with a single purified antigen or a peptide, or more of a gamish, a large set of extract from cells, or putting together multiple epitopes or multiple antigens? And the schematic really is designed just to kind of show, at least in theory, how one might be able to view and balance this idea of, the response rates that one might get, or, which is simply just the, kind of the aggressive cytoreduction equivalent for these immunotherapies, how that kind of might vary from dealing with something that is, very broad acting in terms of its range of antigens or immunogens versus something that might be more specific. Now, of course, at the end of the day, we know that it's been unclear, because there are too few historical precedents to point to, at the end of the day, whether it really will be better. Certainly there are cases where it may be easier, but it's unclear whether it really will be better to end up with, a single antigen, a set of defined antigens, a large unknown gamish of antigens, in the case of even a Provenge, where we have the antigen presumably being passed, because that's what the autologous dendritic cells are pulsed with. It's not even clear that that's really the driver of the activity in those patients that do have a response and do have a benefit. So a lot of complexity to this idea, but nonetheless, the hope, at least among many oncologists and also experts in immunotherapy, that might be a broader type of approach and get around some of these limitations of the more focused mutation or aberration targeted types of small molecule or other approaches. So the times there are changing from Bob Dylan. So this, over the past 2 years, I think it's been really fun for those of us who have been long hoping for and long believing in the power and the ultimate success of immunotherapy. But I think it's also, standing back, it's been, an amazing sea change to, basically go from really being derided, and, I use the word pariah quite seriously, because no major biopharmaceutical company that goes from large biotech on up that we were involved with over, say, the past 5 to 8 years, when we were doing opportunity searches and helping them to identify prospective in-licensing or M&A opportunities, wanted to look at anything having to do with immunotherapy. Now, of course, that was when mostly what they had to choose from were things based on cell lines, things that were autologous in one way or another, things that had more complexity, more challenges in QC, et cetera. And so it is not surprising that the minute one had something that fit neatly in the box of what industry likes, meaning a monoclonal antibody, the tried and true proven blockbusters within oncology, let alone elsewhere, but to be able to say, oh, not only is this an approach that we are comfortable with, finally, though it took 30-plus years to get there, but also an approach that has significant and sometimes surprising activity, as we'll talk about a bit more. So both last year and at this year's ASCO, it really has been the story of these immunotherapies, whether CTLA-4 from BMS or now the PD-1s more recently from BMS, Merck, and others that are working in the space as well, have again kind of continued to fuel this interest because not only do they have significant activity, oftentimes single-agent activity, but because, again, it fits neatly in this easy worldview of the antibody and something that is well-known and not difficult to deal with. And, of course, the Wall Street itself is no longer as skeptical as they had been. And, you know, I had only kind of recently kind of stumbled across this Citibank perspective. But this is, you know, as often happens, maybe this is going a little the other way. But nonetheless, you know, the title says beginning of the end for cancer. Well, I'm not sure I would go that far. But from the idea of kind of grabbing your attention and saying something has changed significantly in oncology and in the industry, you know, that title certainly does it, and kind of their projections, you know, how valid or not is not as relevant as the fact that it is now on the radar screen. It is now recognized as being a significant approach. I want to underscore that approach, and we'll talk about it more because the idea of approach makes it sound like it's a monolith of approach, and it's not. It's a very diverse range of approaches, and we'll discuss that briefly. And literally, you know, just a day or two ago, I guess this was just yesterday, you know, new companies are starting up, whether they are spun out of academia in many cases. You know, there's a lot of hoopla here. This is a very nice kind of multi-major center collaboration with, of course, the role now of the very well-known Hans Bishop. So Juno Therapeutics is now kind of the latest entity just spun out, taking advantage, in this case, of the idea of engineering smart, what they call smart T cells, and we'll talk more about that in a little bit. But clearly, the willingness of VCs and others to fund in this space now represents kind of a new generation and a new worldview about this, which is, you know, a big change from the days when many of these biotech stocks that were in gene therapies, cell therapies, immune therapies, and all of those overlapped to varying degrees were tremendously pounded. And a lot of their fortunes did start to rise when you go back to the days of Dendreon. So, you know, regardless of what may feel about the true validity of that product, nonetheless, they did affect a significant sea change, and shortly thereafter, as did ipilimumab. So we'll touch on that in this quick historical overview. So I think it's important just to remember where we were, because to get to those two big, you know, fierce biotech headlines of, you know, immunotherapies are ruling the roost from, you know, the 2012 ASCO and this year's ASCO, that it was not that long ago. So, you know, whether it's you go back 5 or 7, 8 years to you know, a very different story where, you know, we had kind of a lot of early buzz in the space and a lot of activity, and then in quick succession a lot of failures, a lot of you know, almost pounding on a bruise. It was rather painful when one was kind of watching from the outside, but particularly when you see things like the CancerVax deal in December of 2004, which then, you know, had blown up by spring and autumn of the following year. So this is just a quick perusal through, and thanks to BioCentury for some of this archival material. But you know, and I'm not going to go through this other to say that just many companies that, you know, in many cases no longer exist or have morphed into other forms, you know, were kind of the early purveyors in this space, and frankly, you know, they laid their lives down in many cases because it was all their failures and the learnings from those that have enabled the increasing understanding, along with what was going on clearly within academia and research there in terms of the immune system, that would ultimately enable both the Provenges, but also the ipilimumabs and the anti-PD-1s. And in fact, probably somewhere in here is still BioVest, but that original, you know, anti-idiotype NHL work and goes back to the days of Larry Kwok, and was started, frankly, early in, I think, some point in the 90s. So I mean, there's a 15, 20-year history behind many of these programs. But as I said, many of them have since hit major road bumps or kind of gone off the map, whether or not the programs were valid or not, but their runways often ran out before one could really prove the validity. But the sea change started to occur with a lot of excitement, perhaps overexcitement, with the box on the right when Provenge gets its approval. And then, of course, it gets a real booster immunization, if you will, with the approval of ipilimumab shortly thereafter, and the acquisition also on the left by Amgen of BioVex, which was kind of a major stake in the ground by a large biopharma entity to say, we believe in conceptually the potential of this type of approach. Now, again, I'm putting this type of approach in quotes because there is no this. It's really these, and I'll get to that very shortly. I do think it is instructive to walk through again, and I have done this once before, but not quite the same way. Going back to a Nature Review 2005, it's not to kind of kick any of these companies or programs when they're down, but simply to show that above and beyond the attrition rates and the risks that we all realize in drug development in general, and certainly very specifically in oncology, that in immuno-oncology it's been I don't have any real statistics. I'm sure someone at Thompson does, or some of the other places, but it seems like it's been a very high attrition rate, and that may be in part because many of these companies were small, did trials on a shoestring, did trials that were single arm, did a number of things, but probably even more so with the fact that, again, there was this learning on the job, not understanding the degree of what one had to do to raise the immune system, whether it was in dosing, absolute dosage, how long the patients had to be followed to see the effect, and we'll get to that in a bit, as well as the combinations, the other signals that are necessary, the danger signals, the co-stimulatory signals. So when you look at this list from 2005, those who have been following us for a while may recognize some of these, but it's sad to say that boom, 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 and and more recently, Vical. There are some that's still kind of kicking in some way. Cell Genesis is not kicking anymore, but now all those programs are in the hands of Aduro with their interesting Listeria program, and a number of other companies have also seen interesting studies with their vaccines when it's realized that patients who had been previously exposed to some of the GVAX vaccines at various trials actually seem to respond better to whatever the new approaches are in that companies are doing. So that was an interesting signal that having combination approaches and kind of priming and boosting was important. So that's not really dead yet. And of course, Oncopage has its Russian approval, and Provenge is continuing to kind of chug along. But if you take a snapshot again from 2009, which is the last time I think that Nature Reviews did this, we have Oncogenics still there and Provenge, but you can see another failure, and I think I also want to highlight here that these are failures on many different types of approaches. If you go back, if I go back to the previous slide, we've got peptide approaches, single antigen approaches, we've got anti-idiotypes, we've got kind of broad cell lines that have been ground up, we've got autologous heat shock like Antigenics, the PAP-loaded autologous dendritic cells. Here we have the anti-idiotype to muck that had failed. Elavectin, as before, was gone. The GP100 part of what BMS was doing, clearly not the CTLA4 part, is gone. And we have a road bump here on Stimuvax, and we'll talk more about that. That's still being pursued. I have a little road bump, speed bump here with GSK's MAGE vaccine. And of course, the BioVax, as I said, still kind of there and chugging along. Not clear what's going on with Trovax, although it seems to not be dead yet, as Monty Python would say. So with that, let's move into a little bit of a closer look at the phase 3 club, and I did this as much to kind of fit this in neatly with an hour. Usually I have a kind of a broader sweep across all of the kind of pipeline activity, but I decided that there were enough interesting programs going on in phase 3. I'm not going to go into super granular detail on all of these, but I think it does bear walking through briefly and taking a look at each one, because again it highlights a diversity of approaches, it highlights a diversity of clinical development plans, still mostly in the hands of biotech, but also some large players already that are there with partnered programs. So I call this Yes, My Old Schematic again, because this is one that I've used before. I'm sure it can be improved, certainly it can be made more elegant, but I think it still makes the point that I alluded to earlier, that there is not a this approach, it's these approaches that fall under immunotherapy, because there are many, many approaches, whether you segment it by autologous or non-autologous, the idea of vaccines being some type of antigen-based approach, and that can be derived, those single antigen, multiple antigens can be derived from various sources, delivered in various ways. And then, of course, there's the very successful now, and again, this goes back to, again, I think that's fitting within the neat paradigm of antibodies, of immunomodulation, where those, monoclonal antibodies, the checkpoint inhibitors, right now are what's fueling a lot of the enthusiasm, but that is helping float everyone else's boat. So if you do a quick snapshot here, and there are lots of caveats around, doing this kind of quickly with one of these databases, but I think the bottom line is don't get hung up on the exact programs, but when you slice and dice this and throw out some redundancies and things that happen because of the idiosyncrasies of these databases, you get roughly 100 phase 3 oncology compounds, U.S. development, of which about 20% are immunotherapies of some kind. And I was going to pop in a slide on the overall clinical stage program right at the last minute. I thought it would be useful, and I didn't add it in because I went and looked, and it's actually about the same. It's about 20% of overall clinical development is directed towards immunotherapies of some kind. And again, that runs the gamut from immunostimulatory components like next generation, cytokines, interleukins, etc., all the way up through the most complicated of, autologous-based cell process-based types of approaches, and of course, the monoclonal antibodies to various types of regulatory and co-stimulatory elements, etc. So again, comes in many, many flavors, and just like 31 or however many flavors of Baskin-Robbins, many of these, and harkening back to that earlier slide that I showed in terms of, the idea of kind of broad approaches versus narrow antigen approaches, and how that may or may not correlate with response rates, and let alone how that might or might not correlate with outcomes, meaning PFS or survival. Nonetheless, there are lots of different approaches, and as I said before, the success of one or the failure of one does not necessarily, de-risk anything else going on in the field, but it certainly, it either hurts or helps when someone fails or succeeds, even if it's a very diverse system, a peptide being compared to an anti-idiotype or to an oncolytic virus. So there are roughly 20 programs, with some quotes around that. So I have apologies to anyone that I might miss in going through this. Simply used one database, ADIS R&D Insight. Had to make a few judgment calls, again, as to what constitutes something that's in phase 3 versus not. There are some things that are kind of listed that, but in many ways are not really phase 3. One which I'll call out that is not on here, and I debated putting on is Aduro, who I alluded to before. But nonetheless, in this 20, it shows a very diverse range of programs, as I said. So we start out with, the Argos program in RCC, which, again, RCC, as we'll see briefly, kind of makes sense. It's historically been where, like melanoma, where you had seen some activity of immunostimulatory approaches, meaning interferons and interleukins, and therefore, had every reason to believe that it was a quote-unquote immunoresponsive setting, and so it's not surprising that many have tried to pursue RCC as well as melanoma with the various types of immunotherapy approaches. In this case, Argos has what I call, I think in one case, kind of the double autologous approach, right? Because these are autologously harvested dendritic cells that are then being exposed to, autologous tumor RNA that through their particular technology. So, both the tumor antigens ultimately, as well as the immune cells, are coming from the patient. So there's some double autologous handling that has to go on there. And at the end of the day, they are showing some interesting data. They have fast track. So it'll be very interesting to see kind of how, that type of program fares. And once we get more of these programs, hopefully succeeding, we can begin to draw maybe some interesting conclusions or distinctions about the benefits or not of autologous antigens, of multiple antigens versus single antigens, et cetera. We can contrast that kind of double autologous approach, as I call it, with Newlink's hyperacute platform, which is basically kind of turning things into kind of a xenotransplant approach by the beta-gal modification, the alpha-gal modification rather, of the cell lines. So it is an off-the-shelf type of approach. They've shown some very interesting, promising data, and as I'll touch on later, it also has an interesting development because it's in kind of the adjuvant space, and I'm going to get to that a little bit later. But I think I'll just mention here that what you'll see in a number of these is that it's not just going in last line, third line, fourth line, et cetera, into heavily pretreated, beaten up patients with all of these programs. There's a diversity of approaches now that have actually been brought forward. The GSK MAGE program, as I said, unfortunately had some data that didn't show any significant improvement in disease-free survival recently, but they are continuing analysis of the kind of the subgroups with their gene signature, and that's another trend to kind of refer to something I'll touch on a little bit later on another slide, which is many of these, as you'll see, are now starting to have built-in some types of secondary endpoints and stratification around various types of subsets or biomarkers, if you will, whether those are defined by immune responses, like antibody titers, or T-cell responses, or gene signatures, or other ways that the patients are kind of getting sliced and diced either before the fact or in analyses that are happening kind of after the fact. But this is interesting. We started with the double autologous, RNA and dendritic cells, and the xenotransplant allogeneic cells, and now we have the kind of the MAGE peptide approach and the HLA restriction, and it has, of course, the QS21 adjuvant there. And I think it's important to emphasize, and again, I'll talk on this a bit later, about just the trial that they did in going on into earlier stage patients. I think is certainly, very commendable, risky, but very commendable, and really only something that a large pharma in most cases would be able to do, because it's logistically and financially, it's a burden. And of course, then we have Advantage, which is a very different approach. Now we're talking about a TK-loaded herpes virus, and kind of co-administration of the the anti-HSV drugs like valcyclovir. This is given intratumorally, so that also is a difference as opposed to some of these others, which are given kind of either through some type of systemic administration. We have now the BioVest, which you saw on that 2005 Nature Review and the 2009 Nature Review slides, still kicking. I think it has some challenges just inherently because of how the field's changed, given how long this has been in development and the chemo backbone that it's been compared against, and how treatment has kind of changed between CHOP and certainly Rituxan, and then Treanda with Rituxan. So kind of interpreting the relevance of PACE with this anti-idiotype is difficult, but again, it highlights the diversity again of approaches with this anti-idiotype idea, and we'll see what happens. Then we get to Emanix with their peptide vaccines, and now it is multiple peptides coming out of their ex-president platform, and I think it's very interesting type of approach. It's a first-line study in RCC, so again, going after the presumptive immunoresponsive setting, and another diverse type of approach with the multiple vaccines, multiple peptides covering a range of major HLA types you can see listed there. Of course, we have the PD-1 program from Merck. I'm not going to dwell on that, but clearly very significant data recently released and very impressive. And so, it'll be an interesting horse race between Merck and BMS, although GSK and Genentech, Roche, and others have programs in this space. I'll jump to now, this the polynoma, which is another in malignant melanoma, which is the shed antigen vaccine. So a slightly different idea than just taking all the number of different melanoma cell lines and putting them in the blender and using that as your vaccination, but using the shed antigens instead. So we'll be watching that. They've shown some interesting positive signals. This is also given intradermally, which inherently you'll see a number of these programs have intradermal administration, which is a well-known, not surprisingly, common for prophylactic infectious disease vaccines as well, for really getting access to a range of APCs. And so we'll keep a watch on polynoma. We'll jump down to the new vax here now in the hand. Well, I guess RXI announced Galena to take the immunotherapy separate from the RNAi work. So this is interesting as much for the fact it's a peptide targeting, the at least when you at first glance the highly competitive space of going after HER2, which now has 3 agents, in the hands of Genentech, Roche. But the idea here is that this is being studied in patients who have low to intermediate HER2 expression, so those that are, not the ones that are typically going to be considered candidates or considered to be very responsive to an anti-HER2 approach, so AIC 1 or 2 plus. And so it's a bit of an interesting idea that they're pursuing with this peptide approach. It's using GM-CSF, and actually, many of the other ones that I've flipped through are also using GM-CSF as part of the immunostimulatory / adjuvant component. This is the first one I think that we've touched on that's in breast cancer. Of course, the BMS's anti-PD-1. We won't dwell on that. I have a slide on that again in a little bit. So, but again, very, very impressive in terms of having not only single-agent activity, but also potential combination activity with CTLA-4. Now we have the oncolytic virus from real-licin, which takes advantage of interesting changes given how predominant RAS mutations are, and the interesting observation of how RAS mutations affect the the antiviral systems that operate within cells, thus making cancer cells perhaps preferentially susceptible to destruction by this reovirus. And so that is being pursued with a lead in head and neck cancer, which is where the BioVex acquired by Amgen had been a lead study, but now the lead of that program is really in melanoma. So that's the first oncolytic virus. It's not carrying GM-CSF or some other immunostimulant. It's working more through its own natural cytolytic activity. Biothera, it's the first time we see CRC. The program's also being developed in non-small cell lung cancer. Is very different from any of these others shown to date because it's not antigen-based and it's not an immunomodulatory antibody, but it is an immunomodulatory, polymer derived from yeast glucan that seems to have some stimulation of various immune cells, including neutrophils and others, and seems to boost responses to antibodies. So it's kind of an adjunctive immunostimulatory approach for specific use, along with extant antibody-based approaches. Of course, those would be antibody-based approaches that are targeting cell surface molecules, where you want to get souped up at ADCC or other types of activity. So, that could be a Rituxan or an Erbitux, et cetera, less likely to be, an Avastin. We have the Bavarian Nordic Prosvac. Again, prostate is one that shows up another number of times, and this is an interesting one because it's a combined system of prime and boost with vaccinia and foulpox, plus the triad of ICAM and B7-1 and LF3, and also GM-CSF in there for trying to provide all the danger and co-stimulatory signals that are going on. So that's a variant on the kind of a viral-based approach, kind of a more closely allied in some ways, if you will, to kind of classic infectious disease type of vaccine prime and boosting response, which is taking advantage of there. And then we have the another peptide approach with the Celldex one, and first time now brain cancer showing up with glioblastoma, with a very different type of idea by going after the V3 variant of EGFR, which has been correlated with worse prognosis. And so, we wish them luck. They have had some positive data. Long ongoing studies continue with both frontline and with recurrent GBM, and I'll touch on that again shortly. We have, as I said before, the BioVax Amgen program. We have Biomira, which I alluded to before, the Merck KGA, Stimuvax, which did hit a bump recently, but now has not been put on hold. But I think some of the articles call it kind of doubling down, and is looking into kind of predefined subgroups where there was seen some more significant differential in survival. Many of these I haven't noticed, but they're in the slides. Have fast track. A number of them have SPAs. A number of them have orphan agreements, U.S. and or EU. So at the end of the day, that roughly 20 phase 3 programs. Here's the segmentation. You can see, not surprisingly, that more are being followed in melanoma, and then renal cell, pancreatic has a fair amount. Not one you would a priori think would be immunoresponsive, but it does have some unique settings and situations that allow different type of development path in terms of adjuvant. The breakdown is only 20 in terms of autologous versus non-autologous, but clearly the vast majority that have progressed into phase 3 are non-autologous. That's probably not coincidental. The numbers are not that different from ones when I analyzed the entire pipeline a couple of years ago, so that's about on par. It only starts to change a bit when you have enough of a sample and can parse it into what biotech is doing, which is versus what large pharma is doing, and there you see a somewhat shift with biotech being, not surprisingly, more willing to deal with autologous types of systems of one sort or another, and biopharma leaning the other direction with any of their internal programs. So, immunotherapy continues to be a rich source of approaches. There are a lot of things to pick and choose from. If one were an investor, if one's a prospective buyer, whether of a program or a company, there's still a lot of traction and attraction in just the antibody-based approaches to targeting the various immunostimulatory and immunosuppressive regulatory cell surface and other targets, the OX40s, the CD137s, I already mentioned B7H3, obviously. PD1 is presumed by many to kind of be the next immunotherapeutic blockbuster in the space, probably indications and potentially in the hands of several different players, although, as we'll get to shortly, this may be where having ownership of more than one type of platform and the ability that that enables in terms of combinations and kind of sewing up kind of an immuno-oncology franchise could be very significant. And I think you can see with this idea of combination too, whether it's with other immunotherapies or other approaches, the ipilimumab's combination studies going on here across a range of different approaches, including chemotherapy, which historically had been considered very unlikely to be able to be combined with immunotherapy, but now is being seen in many cases to provide the appropriate cell death and necrotic and setting to soup up the immune system. It's really all in the timing, perhaps, if you will, and then many combinations with targeted agents, which in and of themselves also are being seen to have immunoregulatory activity like Revlimid, like sorafenib, et cetera. So, as again, not surprisingly, there's a lot of activity and interest in the checkpoints, both the ones that are more well described, so that kind of the chase that's happening in PD-1, as I said, between BMS and Merck and maybe GSK and Roche Genentech and others. But also you see some people, such as the Bayer deal with Compugen, around early stage discovery of novel checkpoints. So Compugen has a platform to identify through in silico analysis, yet to be described entities that they then can actually then find and prove actually do exist. And so they did so and predicted and then found additional checkpoint inhibitors, and that's what the Bayer deal is on. Or programs that have kind of been around in the hands of others, like the CD40 agonists, and also been looked at with antagonists. But now, like with companies like Apexagen, are trying to bring that program forward again, not only because it has immunomodulatory activity, but also maybe some direct cell-killing pro-apoptotic activity. So a lot of interest again in the antibody approaches, 41BB. I already mentioned 137, so there's a whole range of those. So FitNitly. So you can kind of compare and contrast that type of approach with a number of others. Clearly, these are likely to have benefit when combined, and various preclinical and early human studies are showing the value of combining CTLA-4 and the PD-1, hopefully with manageable tolerability and toxicity. But you can compare and contrast that type of approach, the antibody approach, the easy off-the-shelf given antibody, with something that's a little more labor-intensive, a little more intellectually distinct, such as the chimeric antigen receptors, the CARTs, the TARs, the smart T cells, as Juno referred to them in their founding press release. There's a lot of generations have progressed over the past few years, more understanding of the various intracellular signaling that has to happen, and the tails of the T cell receptors are getting longer, and more kind of co-stimulatory components are being involved with this, but still it does involve some extensive engineering of patient cells in order to then provide. But on the other hand, this, rather than saying a priori, all that engineering takes a lot of time, it's going to be too costly, that certainly would be true if one was talking about, adding 2 weeks or even adding several months of life. It'd be hard to envision how one would justify that. But the data that's been seen to date, at least in the case of ALL, through various groups, including the Memorial Sloan Kettering group on the left, or the and Penn group of Carl June, on the right, is that these have had very dramatic efficacy, getting to minimal residual disease in difficult ALL patients. There have been some tolerability issues and some interesting application of approved anti-IL-6 agents in terms of kind of managing some of that. But nonetheless, it represents a very interesting, hot new area. A number of companies have moved into the space: Celgene, Bluebird, Baylor, and academia doing spin-outs and collaboration. So it will be interesting to see how this field progresses. It's one thing to envision the applicability in heme malignancies, but even there, the activity has been much greater, say, in ALL than it has in CLL. But then broadening that out to solid tumors starts to get a little more complex. And so the real challenges for these, less so in the complex and interesting engineering of the next generation CARTs than it is in identifying appropriate tumor-specific antigens or suitable tumor-associated antigens that are going to have the right therapeutic window of sufficient activity and limited damage to host tissues so that the tolerability will be and the safety in AEs will be acceptable. I think it's also interesting in terms of just those phase 3 programs and others to note that it's not just these the easy cancers, as I said, like RCC or melanoma, easy in the sense that there was some precedence and belief that they're immunoresponsive, but it's the tough ones like lung cancer, which we're really at the bottom of most people's list historically, and no one was really thinking of going. But some of the data that's been presented on the PD-1 agents from BMS and others in the space is pretty impressive, and it'll be interesting to follow that as those proceed, particularly how they might be combined with a standard of care or with other immunotherapies. And it also highlights this idea that I alluded to before with a number of these phase 3 agents and many of the other agents, which is a trend in oncology right now. It's the stratification and the biomarker group. So there are observations about patients that are expressing PD-L1, and some studies, some of the studies are actually ongoing to kind of track and see how patients respond in terms of their expression of PD-L1 versus not, which makes it somewhat more similar to what's going on with some of the other drug development outside of immunotherapy in oncology. But just looking at the 20 programs that I listed, at least from what I could see that's in the databases, the majority still don't have a biomarker, or at least it's not indicated. That doesn't mean there's not something being tracked. And 8 in some way have some reference to either a biomarker or some stratification based on antibody titer or T-cell response or something. So this convergence of kind of that broad-acting, and to some degree you might say it's a little bit contradictory, the broad-acting immunotherapy and the stratification idea, that's kind of the more common approach now within, certainly many non-immune mechanisms in oncology, and kind of now overlapping these two. And there's lots of work going on. Axel Hoos, had argued for this for a while and has helped kind of move this along with many others of being able to try and harmonize and look at the various types of criteria, immunosuppressive criteria, so that you can have some early readouts. There are a number of different profiles. This is one just came out, very recently that may be relevant, and this one seems to be able to be predictive in terms of stratification and prognosis. But at the end of the day, whether it's this or something else, we are seeing a convergence between, immunotherapy now and the kind of stratification or personalized precision medicine elements, which makes a lot of sense. Again, going back to the idea of needing to have sufficient benefit, not just incremental benefit, at the price that one wants to charge, being able to have a more defined group that is going to be more likely to respond or more definitive to respond, whether it's through an HLA subset like Ematics, whether it's through some other type of segmentation through a gene signature like GSK's MAGE system or something else. At the end of the day, as I said, it's going to take these combination approaches probably to be meaningful, and this has some relation also to the kind of the delayed effect, the tail effect that's seen, and sometimes the disconnect between PFS and OS, or certainly between response rates and outcomes with immunotherapy. And that has some important implications in terms of how one thinks about doing combinations and the types of studies that one does. And in fact, one of the interesting observations that I already alluded to about that phase 3 club is they're not all, last line salvage therapies or even monotherapies. They're being looked at lots of different ways. They're being combined with standard of care chemo agents in adjuvant setting, like the Nulic approach for hyperacute pancreas, or that's being combined with a targeted agent, meaning sunitinib in first line in RCC. A few years ago, I don't think people would have envisioned that someone would be moving these immunotherapies first line, despite the fact that everyone was saying that's where you need to go. Maybe many of those programs that failed before failed because they were in later lines of therapy, harder patients. Maybe their immune systems were compromised to some degree. So adjuvant settings, first-line settings, minimal tumor burden or minimal residual disease settings make a lot of sense for what are typically not kind of massively destructive cytoreductive type of approaches, and also that take longer to work. The Nuvax, which I said is going for the low to intermediate HER2 expressors, or Celdex, which is going first line, as well as looking at recurrent patients. So people are getting past and moving on and looking at other areas. It'll be interesting to see immunotherapies in kind of maintenance types of situations. And of course, as I said, the combination approaches are critical because there are so many different cell types that one wants to engage in order to really get the most bang for your buck with the immunotherapy. Having multiple platforms and modalities in your own franchise, as I alluded to before, can certainly be potentially very powerful, and it will be interesting to see when BMS has their PD-1 approved, if Merck has theirs approved, and if data is really strong in terms of combining with anti-CTLA-4, does that mean that BMS has kind of sewn that world up because they own both, or what the implications of that remain to be seen. But certainly the idea of being able to control those assets and do the types of studies to optimize the combinations and optimize the therapeutic window is something that one ideally would like to be able to do. I point out just one kind of set of recent deals really have happened over the past year here with Roche, who's kind of starting to make a play in the space, most recently with their Maddox deal, going for not the advanced stage program in RCC, but earlier staged vaccines across a range of different tumor types. Their deal with Immunocore around the T-cell approaches, so peptide T-cells, and the Inovio, which is really the single antigen, the PSA or PSMA type of approach. So, it'll be interesting to see, and those are all kind of early stage deals. So a number of early stage deals spreading the bets across different types of immunotherapy platforms. Maybe that's the way to go. And just in conclusion, this idea of being able to do these combinations, whether it's PD-1 and the CTLA-4 or any other combinations, being able to perhaps not only move the curve and affect earlier, so to get a response earlier and maybe a more dramatic response earlier, but also to extend the duration and to extend the kind of the tail end effect for these patients, may provide a very significant new place where immunotherapies will be able to go and make significant, innovation in terms of management in cancer. So with that, I thank you for your attention. Unfortunately, I left only about a minute for Q&A, but I thank you for your attendance. And if you do have any questions, please feel free to send them on to us, and we'll answer them within the next day or two to the best of our ability. So I thank you for your attention.
Thank you, everyone, for attending. We do have a few questions, Jeff, and I think in the interest of time, we'll answer them offline if that's all right with you.
Sure.
Okay, thank you very much.
All right, thank you.